What is a gene? The human body is made up of an estimated 30 trillion cells, with about 200 different types present. Each of these cell types are responsible for different functions in the body. Every cell has DNA located within its nucleus. The DNA codes for genes, which make up the 23 pairs of chromosomes present in humans. How is a gene expressed? Genes contain the information needed to make functional proteins. The transition from gene to protein occurs in two major steps: transcription and translation. This process is known as gene expression. First, the DNA goes through transcription. This process is regulated by small proteins known as transcription factors. The transcription factors bind to the DNA, and based on their binding position, the cell controls which genes should be expressed. Next is translation. When the mRNA from transcription reaches a ribosome, the mRNA sequence is read in sets of 3 bases known as codons. These codons code for specific amino acids, which are used as the building blocks for making proteins. The specific genes expressed in a cell determines the proteins that are made, giving each cell type its unique function. So what is direct reprogramming of cells? In the last decade, science has seen major breakthroughs that use transcription factors to reprogram cell identities from existing cells. The idea comes from using human embryonic stem cells, HESCs, that are isolated from blastocysts, the early stage embryos. Due to the fact that these HESCs are isolated in the early stages of development, they can become any type of common cell type. These reprogrammed cells are known as induced pluripotent stem cells, iPSCs. By mimicking the transcription factors found in HESCs, researchers can revert fully mature adult cells back into HESCs and then back into any cell type. What sets apart direct reprogramming from iPSCs is that direct reprogramming doesn't require the use of an iPSC as a go-between. If the right group of transcription factors are introduced into the target cell, then theoretically the cell can reprogram itself into the desired cell line. What does this mean for the future of regenerative medicine? Patients could potentially have some of their own cells removed and directly changed into the cell type that they need. These cells could be easily obtained through something as simple as a blood sample or skin biopsy. Since these cells come from the patient, it could eliminate the possibility of immune system rejection as seen with organ transplants. When the body recognizes something foreign, its response is to eliminate it. Even with anti-rejection drugs, this is still a risk for transplant patients. Future implications of direct reprogramming creates the possibility of regenerating injured tissue and even organs. By introducing the necessary cells back into the body, it would have the capability to grow new organs, drastically reducing the need for transplants. This would revolutionize regenerative medicine. Direct reprogramming of cells is our future.